less frequently. Those mistakes will be in important regions of the genome. And that will lead to a mutant RNA, which then leads to diseased protein, which then leads to either no function or a gain of a new function that is problematic. So the whole concept of gene therapy is to go back and interfere with the very first part, fix the very original portion of the disease. Instead of trying to, fix the disease protein, you go all the way back and fix the mutated DNA. And so how many genes are we trying to fix with this? So the first gene therapies you might think of are for some common genetic diseases. You might think of hemophilia, cystic fibrosis, Duchenne muscular dystrophy. Make it pretty bad. Turn off video. Good idea. Yeah, I am at a hotel, but I paid for the extra Wi-Fi, so I was hoping it'd be better. Video. Stop video. There we go. Same. All right. Hopefully that gets a little bit better. I apologize if I'm breaking up a little bit. Okay, so the video helps. That's good. All right. So if you missed something, we are trying to correct genetic disease by delivering the broken piece of DNA. So here is a schematic of delivering a missing gene with a virus. Often we use this virus called adeno-associated virus. It's a very small, very non-pathogenic, very not harmful virus. And so you might think of some of the common genetic diseases, and some of those are labeled here. Duchenne muscular dystrophy is on the X chromosome, so is hemophilia, cystic fibrosis. So how many genetic diseases are really on the docket for us to fix? So the Rare Disease Society keeps track of this, and there's about 3,700 genes that are associated with the genetic disease, and that's out of 25,000 genes. So there's a lot of rare diseases. In fact, the Rare Disease Society says 1 in 10 people have a rare disease, and so rare diseases in aggregate are actually not rare. So what do we need to do to fix the DNA? There's a few steps we try to think about when we're designing these types of things. The first is we have to find the gene that we need to correct. Human genome is humongous. It's 3 billion bases long. So how do we find it in that mess of DNA? Number 2 is we need to make the desired modification at that location, but only in that location. We don't want to edit other genes and cause something even worse to occur. And hurdle number 3 is we need to deliver the technology that does number 1 and 2. We need to get to the right cells in the body. So if it's a disease of the brain, we need to get into the neurons in the brain. If it's muscle, you need to get all of the muscle fixed. So how do we get these technologies to the right cells? And so researchers in this space will work on all 3 of these hurdles. So, all right, so let's work on that first question first. The genome has 3 billion bases. How do we begin to target one specific part? Okay, so we kind of try to think about this in terms of zip codes, genomic zip codes. How much information do you need to know about the genome in order to, or the spot that you want to edit to fix it? So with 3 billion bases, the math turns out to be roughly 20 bases of information. It gives you enough information to identify a unique spot in the genome. And so all of these technologies I'll describe here will do the same thing. They'll target about 20 bases of information. So the first technology that really started the field of gene editing is this meganuclease. It is a protein derived from yeast. There's other organisms that have these too. They are entirely made out of protein, and they are very difficult to engineer, but if you can make one, it will go and localize on a stretch of DNA that you design. Technology that made the meganuclease idea a lot easier is this idea of zinc finger nucleases. So these are little proteins, and the proteins are designed in a way that they bind to sets of 3 bases of DNA. They're derived from human proteins, and you string them together in these little arrays, and then you can get specific targeting of certain sequences. So they go into the cell nucleus and they search around and they find different sequences of DNA. This is what the field was using for a long time. until in about 2011, a new technology arrived called TALENs. TALENs are derived from a plant pathogen, and these bind to single bases of DNA. So they're little proteins that stick to single bases of DNA. So you could string these in an array. You could say, I want a T, I want an A, and you could put them together in an array that will stick to DNA. But TALENs would probably be used a lot more commonly if it weren't for CRISPR described the next year, which sort of made gene editing so much easier, democratized the field of gene editing, and made this research field explode. So we'll talk about CRISPR. Now, what is CRISPR? Where did CRISPR come from? It certainly isn't a tool that someone made to do gene editing. It's a natural, actually occurring phenomenon. Okay, so I hope that we're all a little more familiar with how our adaptive immune system works. So we get exposed to a virus or a vaccine, our body mounts an immune response to that vaccine or the virus. And so the next time we see it, we destroy it much faster and we're much less likely to get sick. So that's our adaptive immune system. So it's adapting to new pieces of information. And so, but we're large, complex, multicellular organisms, and we have these complex immune systems. Bacteria also have an adaptive immune system. And that really surprised me when I first learned about this, that bacteria can have an adaptive immune system, meaning they can learn from their environments and develop an immune system. So bacteria and bacteriophage have been fighting this eons long battle. Bacteriophage are viruses shown up in the right that infect bacteria, take over their machinery and make more of themselves. So when bacteriophage infect a bacteria, the bacteria will likely die. But bacteria that can incorporate a little bit of information about that bacteriophage into its own genome can remember that bacteriophage for next time. And when that bacteriophage shows up again the next time, it can fight it off and kill it without the bacteria cell dying. And so in the first phase, the bacteriophage shows up, injects its viral DNA, and this bacteria cell gets very lucky. Somehow it manages to survive the initial infection and incorporates that little bit of information in this spacer sequence into this long array of repeats. So you can see the repeats in the colored hexagons in the bottom right. And this information is stored and remembered. And it's actually a reading history of different bacteriophages that have infected that bacterial cell over time. So it remembers this information. So on the second phase, the bacteriophage shows up and tries to infect the bacterial cell. The bacteria responds by creating this new enzyme with a new CRISPR RNA. So this enzyme Cas9 and these RNAs go together, find the sequence of DNA that is infecting them, and cut it in half and destroy that bacteriophage DNA so it can't copy itself. Okay, so this was a profound discovery. We won the Nobel Prize 2 years ago for this discovery to two scientists, one Emmanuel Charpentier and Jennifer Doudna, for really working out the mechanism for how Cas9, this is the protein, and the guide RNA, those two together make the CRISPR system, how they work. So one thing remains, though, from here is, so this Cas9 breaks DNA. It kills bacteriophage. How do you use it to fix DNA sequences? So that leads us to our second question. All right. CRISPR is amazing. It uses an RNA sequence to find the right sequence of DNA. So let's go back and show that figure one more time. So down at the bottom, you can see how CRISPR is structured to work. You have this protein, this Cas9 protein, and then red is the RNA. And the RNA is what you use to program where you want to go in the genome. So you basically type in the sequence of RNA you want. You order that. You have the Cas9 protein. You have this new RNA. You inject that into cells, and it will go and find that sequence, and it will cut that sequence. And it does it at a predefined location every time. And so it's very reproducible, but it cuts the genomic DNA. That is not a way to repair a genetic disease. So how do we use the idea of breaking DNA to repair a genetic disease? All right, so that comes out of a new field, another field of DNA repair. And so this was another Nobel Prize. and probably another one coming along shortly. So there are lots of different ways our cells respond to errors in our DNA. So our DNA is constantly being bombarded. You go outside, you get some ultraviolet radiation, you go on a plane flight or eat a banana, we're constantly getting radiation destroying our DNA. And so if we didn't have a way to repair that was already present in our cells, multicellular life couldn't exist. So organisms have these ways of repairing DNA. Lots of different pathways exist to fix DNA. The one that I want to point out, though, it actually was not a subject of the Nobel Prize, is homologous recombination. So we can use this DNA repair, that human cells already have in them, and fix sequences of DNA after we break them. So how it works is you use something that breaks the DNA. In our case, that's going to be CRISPR. It can be a lot of different things. So you use CRISPR to break a sequence of DNA. Now it's up to the cell to repair it. And if you're clever about it, you can make the cell repair it to the way you want it to be. So one example of that is to use a double-strand break. So the DNA is broken in half by CRISPR, and then you don't supply anything. The cell will repair it by this pathway called non-homologous end joining. That's NHEJ, and that makes mistakes when it repairs. It tries its best to repair it and often does a really good job, but sometimes it leaves small mistakes, which you can use that to break specific genes. You can also do 2 simultaneous double-strand breaks and delete portions of the genome out. So say you had a gene that was causing a problem. It was a gain-of-function protein that caused toxicity. You could cut that gene out and it would no longer cause that problem. In some cases, like cystic fibrosis and sickle cell, you really need to get the gene back to the way it's supposed to be. You have to repair it. That gene is necessary and we have to repair it. So how do you do that? So we can use a double-strand break and we'll supply a homologous template of DNA. So that homologous template, this works, our genomes are diploid. That means we have 2 copies of each chromosome. So, we have 23 chromosomes, but really we have 46 in each cell. And if a gene is broken in one cell, it will go and find that second chromosome and pull it over and use that as a template to repair the missing information. We can trick the cell into doing that same thing with a sequence that we provide. So you can provide the sequence that's now corrected and it will incorporate that new sequence. And so that's how we can repair precise mutations. And there's a lot of interesting things outside of this that you can do, kind of outside the box applications. You can use CRISPR for all sorts of other things. And I'll show a short video on that I think visually explains that better. Someone asked interesting question, can bacteria share viral DNA sequences with other bacteria, or is the resistance only passed on to progeny? So there is mechanisms for horizontal transfer, but that would be really unusual. I think most likely it's just shared with their descendants. So the ones that are the lucky ones that survive the initial infection phase will go on to propagate and spread that information that way. That's a really good question, though. And there's some other mechanisms. I won't talk about those, but I will skip over to this video. This video does a better job visually of showing what's going on than I can probably describe. So I'll show this video and then we'll continue our discussion.
The CRISPR-Cas9 system is a tool for cutting DNA at a specifically targeted location. The technique has already revolutionized gene editing, but scientists are always looking for new possibilities. So what else can CRISPR do? Since being discovered in a bacterial immune system, CRISPR-Cas9 has been adapted into a powerful tool for genomic research. There are 2 components to the system: a DNA cutting protein called Cas9 and an RNA molecule known as the guide RNA. Bound together, they form a complex that can identify and cut specific sections of DNA. First, Cas9 has to locate and bind to a common sequence in the genome called a PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes. But making double-strand breaks isn't all CRISPR can do. Some researchers are deactivating one or both of Cas9's cutting domains and fusing new enzymes onto the protein. Cas9 can then be used to transport those enzymes to a specific DNA sequence. In one example, Cas9 is fused to an enzyme, a deaminase, which mutates specific DNA bases, eventually replacing cytidine with thymidine. This kind of precise gene editing means you could turn a disease-causing mutation into a healthy version of the gene, or introduce a stop codon at a specific place. But it's not all about gene editing. Several labs have been working on ways to use CRISPR to promote gene transcription. They do this by deactivating Cas9 completely, so it can no longer cut DNA. Instead, transcriptional activators are added to the Cas9 by either fusing them directly or via a string of peptides. Alternatively, the activators can be recruited to the guide RNA instead. These activators recruit the cell's transcription machinery, bringing RNA polymerase and other factors to the target, and increasing transcription of that gene. The same principle applies to gene silencing. A CRAB domain fused to the Cas9 inactivates transcription by recruiting more factors that physically block the gene. A more outside-the-box idea for using CRISPR is to attach fluorescent proteins to the complex, so you can see where particular DNA sequences are found in the cell. This could be useful for things like visualizing the 3D architecture of the genome, or to paint an entire chromosome and follow its position in the nucleus. CRISPR has already changed the face of research, but these new ideas show that what's been achieved so far could just be the tip of the iceberg when it comes to CRISPR's potential. Whatever comes next, it seems the CRISPR revolution is far from over.
All right, so but got some interesting questions during the video. So what happens in a case where the double-strand break repair process doesn't introduce errors? Nothing. So that's a good question. So that would then still express the gene, but an interesting thing that happens, I don't know if I can show, go back to the YouTube video, is I have a picture. So I'll just describe it. So if that happens, that reconstructs the CRISPR site again, and it will cut it again. So it can keep cutting until it actually makes an error. And actually, there's some evidence that that's what happens. Is 99% of the time that repair pathway works perfectly, so right back the way it was, and CRISPR just cuts it again. Another question was, where does the DNA come from? Where do the bases come from that the DNA is using to resynthesize itself or to make RNA? And we have whole pathways that synthesize those. There's this extra nucleic acid, predecessors floating around in the nucleus, ready to be used. So that's important in, DNA replication, RNA transcription, et cetera. So, all right, and can mutations cause dangerous side effects? And that is a big concern. Not so much at the target site that you're aiming at, but what if you made it? So there's some places in the genome that look similar to other places. Could you accidentally edit a different spot in the genome and cause some sort of problem? Cancer is obviously the biggest concern for that. And so some other questions following up on that is the goal to edit every cell's DNA or just some threshold? And that's a very good question, very disease specific question. There's some diseases, like imagine you wanted to fix a cancer, like a tumor. You would have thousands, tens of thousands, millions of cells if you corrected 99% of them, that's great. That might regress the tumor, but the 1% that you didn't fix are still there and will regrow. So, that could be one where you have a high threshold. In some genetic diseases, having a small threshold will probably repair. And we'll share an example of that in a minute, where as little as 5% might be therapeutic. And nutrition, there's a really interesting thought on that, that your genetic code is like 15% predictive of your longevity, and your environment is like 85%. So like which one would you know more about somebody? Would you know more about somebody from their genetic code or their zip code? And that kind of gets outside of my area of expertise, but you would know more about somebody's health and longevity by their zip code than their genetic code. And then of course, when I'm talking about monogenic disorders, that's sort of the exception to that rule. So one of the questions is, how do we get these gene editors to every cell in the body or every cell that is diseased in the body? And so there's several different delivery approaches that you might think of. We famously talked about the one on the right, embryo editing. Now that certainly fixes every cell in the body if you could fix it, because every cell will then be, from that original cell. But what if the person's already born? What are we going to do then? Obviously, if you're already born, you're not a candidate for embryo editing. So we're looking at doing some other delivery mechanism. One option is to take cells out of the body, fix them, and then put them back in. So we call that cell therapy. It's still a type of gene therapy in that, we're fixing genes, but we take cells out, fix them, and put them back. So that is usually, we're thinking about disorders of cells that can be removed. So sickle cell, for example, is blood cells. So we can remove those, fix them, and put them back. Whereas a skeletal muscle disorder, on the other hand, thinking about taking out your skeletal muscle, fixing it, and putting it back is more problematic. Gene therapy or gene delivery to the actual human is another way of doing that. So in that case, we often would use a virus to encode the information that we need to fix the gene and then infuse that virus into the patient. And there are a couple of clinical examples of that. So when we were thinking about this process, we kind of think, why would we do a certain way? So why do it ex vivo? Like if you can take cells out, you can edit them, you can screen them extensively for issues before you put them into a patient. So if something went wrong with the editing process, you would know in advance. If you need to edit in vivo, so in vivo, we're talking about editing the tissues inside the body. So editing the brain, editing the muscle, why would you need to do that? You have to, because you can't pull the tissues out. The third one, why edit the embryo? That is only during in vitro fertilization. That's the only way we can do it right now. And there's advantages and disadvantages to each. And certain applications are, going to be more appropriate for some diseases than others. Okay, so now let's get back to Dr. Hu's experiment. We pronounce that Hu. It looks like he. It's really confusing because it's like in articles, it's like he did this. Anyway, so Dr. Hu's experiment. Okay, so first we need to talk about Timothy Ray Brown. Does anyone know who Timothy Ray Brown is and why he's famous? So Timothy Ray Brown is a man who was HIV positive. Then on top of that, also developed leukemia. So only want to be clear about this is when I talk about an HIV cure, this is not an HIV cure. Even though this man was the first man to be cured of HIV. But the thing that happened to him is not a cure for HIV in general. So what happened is he developed leukemia, and he was with a smart group of doctors who were studying this protein called CCR5. And CCR5 was thought to be the protein that HIV stuck to get into T cells, get into your white blood cells, and cause the disease, cause AIDS. So his doctor decided when you have leukemia, you have to kind of clear out your bone marrow cells and get a bone marrow transplant. He thought, why don't I take a bone marrow transplant? Why don't I get one from someone who's lacking this protein? Then, in theory, his white blood cells would no longer be attacked by his HIV virus. So that's exactly what they did. So they destroyed, well, they didn't have to destroy, the they got it from a patient who already was lacking CCR5. So they got white blood cells from a patient without CCR5, infused those bone marrow cells into Timothy Ray Brown, and that cured his HIV. So he was the first man effectively cured of HIV. So the idea being, if we lose this CCR5 protein, you get resistance to HIV. That gives us an idea. Hey, we can break genes with CRISPR. Why don't we use CRISPR to break the CCR5 gene? And people have been working on that to do that in adults, where they go in and get a bone marrow transplant with CCR5 edited genes. So what are the cons of doing CCR5 editing? It's very unusual that our bodies evolve a protein that has no useful function. So CCR5 probably plays some sort of role. It might make them more susceptible to certain types of viruses, but in general, people lacking this protein, it's not really a genetic disease. They live normal lives. So there could be some cons with doing that, even in adults. Okay, so Dr. Hudd's experiment, he described it, but let's go over to, it was quick though. So there was a male in the experiment who was an HIV carrier and a female that was HIV negative. They went to the clinic and did in vitro fertilization, but with one extra step. They infused Cas9 and a guide RNA, so the CRISPR system, into the embryo. They waited a while for that embryo to grow into some more cells. So in the test tube, they picked off a few cells for pre-implantation genetic diagnosis. So that is where you can pick off a few cells and you can screen them to see what their DNA sequence is. This is commonly used in vitro fertilization to eliminate risks of genetic diseases. So they monitored, after the implantation, they monitored the pregnancy by looking at cell-free DNA. So that's the idea that the baby will shed DNA into the mom's blood and serum. You can actually pick out sequences of DNA. And if any of you are aware of this or have known someone who is having a baby, they can offer you these services now where they can identify the sex of your child. They can identify if it's predisposed to any genetic diseases when you're 12 weeks along, which is, it's crazy that there's enough baby DNA in the mom's blood to be able to do that. Anyway, so then the two twins were born and they did some more sequencing and analysis to figure out if their experiment in fact worked and if it was safe. Anyway, so you guys read a little bit about this. I need to hide this bar. Do that. Okay, so anyway, so you guys read a little bit about the experiment. Is there any thoughts so far on the ethical lapses that may have led to this experiment? I have a few that I can discuss as well. Yeah, so yeah, that's a good question. Ethan, that's a good question. Do you have to deliver directly to the embryo or can you put it in the mother's bloodstream? Now, there is this idea of in utero editing. No one has demonstrated that in people yet. So, but in contrast, right now, the way embryo editing is done is injecting into the single cell embryo during in vitro fertilization. So that still has to occur at the single cell stage. There also is the possibility that editing can occur after the single cell stage. The cell's already divided into two, and now you have 4 copies of the gene to edit instead of just 2. Yeah, so I agree with Emily here that a lot of the ethical issues are kind of outside of my area of expertise. So patient consent was a huge problem. Apparently, what I've heard from people is that it read sort of like an HIV vaccine. You're getting an HIV vaccine for your kids, and less of we're going to modify their genomes. So another question is, can we have an approved human embryo gene editing in the future? And if so, how soon? That is a good question. I don't see it coming soon for a couple of reasons. And one is that there's very few true use cases that exist for doing human embryo editing to actually correct genetic disease. And I'll describe that here shortly. that there's other ways to fix genetic diseases that are easier than that. Oh man, lots of questions. Yeah, so we won't know about the safety of this, for a long time as the kids develop. And yeah, and people without, and it's, science communication is an important topic here because, people who don't have the background in this space may get a very different view. And I've noticed that in some of the talks that I've given, that they have a very different view than the scientists who were engaged in this subject before. Which is really good to be bringing people in to have all these different viewpoints. So, yeah, so those are all good. One thing, so I do feel qualified to discuss some of the ethical lapses that I am kind of outside my scope. But one of them is the genomes. So this was stolen from his slide, and I had to kind of recreate it. All right, so in green, at the very top, it says parents' genome. So this is going to be the genomes of most of us. Most of us are going to look like this at this locus. So this is the CCR5 gene, a small piece of it. So the patient that gave bone marrow to Timothy Ray Brown, he was what they call Delta 32. So you see this Delta 32 means they're missing 32 bases of information, and that makes that protein not work. One thing to remember is when RNA makes protein, is it makes it in groups of 3. So 3 bases of RNA makes a single peptide for amino acid for a protein. That 3 rule is very important. So if you have a mutation that is not divisible by 3, it's much more likely to break the protein than something that is divisible by 3. Because each amino acid that comes off of here is going to be dependent on the amino acids before it. Delta 32 is not divisible by 3. So that means all of that red information there is broken. The cell can no longer read that correctly. Okay, so kid number 1, codenamed Lulu, lost about 5 bases of information. That is not divisible by 3, so this first gene is broken, probably. The second, so remember we have 2 chromosomes for each. So this protein is encoded on a chromosome that, there's 2 copies of it. So to break the whole thing, you got to break them both. So to really remove that protein, you got to break both genes. So the second one is a +1. So +1, also not divisible by 5 or divisible by 3. So therefore also broken. So that's why I have that in red. All right. So now where it gets really curious. Oh, okay. So was this intentional? Were they making these different mutations intentionally? No. So one of the things is that that error-prone process is semi-random. So it makes kind of these weird, sometimes it deletes a few pieces of DNA. Sometimes it inserts a few pieces of DNA. Sometimes it deletes 10,000 bases of DNA. Sometimes it inserts a piece of DNA that might have been floating around. So it's inherently error-prone and semi-random. I've been missing out on some questions here. Okay, I'll get back to those as soon as I finish describing that. So, okay, where it gets really crazy is Nana's genome. So the first allele is not edited at all. So the first copy of her chromosome, it's not edited at all. So she is not immune to HIV, just like someone who is only homozygous for the Delta 32 mutation or heterozygous for the Delta 32 mutation. Nana is just as immune to HIV as the rest of us. So she's not immune to HIV. Then very curiously, there was a copy that has 15 bases missing. So 15 is divisible by 3. So that's why I put this purple instead of red. That is now a new protein that no one has ever seen before. It's missing 5 amino acids. It looks a lot like a CCR5 protein with an important piece missing. So in experiments in mice where a similar error may have occurred, not in this protein, but in a different protein, it can cause immune phenotype changes, including immunodeficiency. So where there's no evidence that occurred in these kids, it could have occurred, and that is risky. So the fact that they chose to move forward with Nana is very remarkable. I saw some comments too. So, that the legal jargon was difficult to understand. can you have I guess there's some kind of Gattaca type stuff, having that kind of control is sort of scary. Even though I think, just to, I think we're a long ways away from that. And yeah, was it necessary to edit these? The kids were not likely to get HIV. So that's one of the important points is when you do in vitro fertilization with an HIV positive parent, you can eliminate the risk of HIV, both to the mother and to the children. So those children were not at risk for HIV. Okay. So some of the other concerns, I think that many of you are bringing up in the comments is a questionable disease choice. Why HIV? The patient consent form was apparently not well written. It was completed in secrecy. One of the big points from the embryo editing consortium was that anyone who wants to do this, like, let's all talk about it. Let's engage the stakeholders. Let's talk with the patient communities before we go forward with this. And this was definitely not the case. It was just a surprise to almost everyone. There were a few people that he did discuss it with before, but very few. And he received a lot of blowback, to put it lightly. Pretty much near universal condemnation. There were a few people who, stood by his side a little, but mostly, some of these are famous people. There's Jennifer Doudna there. Francis Crick Institute is represented. Eric Topol, another person. So there's a lot of ethicists and geneticists, and so that condemn this activity. Okay, so if we all kind of feel, we can talk about this more during our discussion section. If we all feel that was an unethical use, what are ethical uses of genome editing? Someone said he went to prison for 3 years. Yeah, he recently got out. And that's a question for another day, probably, on whether that was the right thing or whether that was the right amount of time. That's a very different question. So, what are ethical uses? When can we do it? When is it necessary? And I agree with Stephanie here. I think, it really looks from the outside perspective like a selfish land grab. You want to be Neil Armstrong putting the flag on the moon for the first time, and you want to be the first one to do something. I don't know if he'll be remembered in history like he hopes he will be. Okay, so let's get to this question first. What cases is gene editing of the embryos the only option? So what diseases might be useful? What about some scientific endeavors might be useful? So one thing is, when we're thinking about embryo editing, we're thinking about a heritable cure, but there's very few diseases that really fit into that category. So I won't go through this whole table, but this is sort of like a decision matrix on when to do certain ones. But an important point, though, is we do have this other competing technology called pre-implantation genetic diagnosis. So assume that you knew two people who wanted to have a child. They were both carriers of cystic fibrosis, and they wanted to have a child without cystic fibrosis. They can go to do in vitro fertilization, select embryos that are lacking the mutated gene, and they can even have a child that does not carry the recessive trait along to the next generation, effectively eliminating cystic fibrosis from their progeny. So with this technology available, and to be clear, to do the CRISPR editing experiment, this isn't like a competing technology in that it's this or this. You have to do all this work for the CRISPR approach anyway. So CRISPR embryo editing is this plus editing. So this is so CRISPR editing is every bit as hard plus more than pre-implantation genetic diagnosis. And so thinking about what genetic diseases fit that category where you would want to fix the embryo. So most, 50% of pregnancies are unplanned. So obviously those children are not candidates for embryo editing. Spontaneous mutations occur, so you did not know. Or most people, like myself, would not know if I was a carrier for a genetic disease. If you don't have a family history, if you didn't already have a child with a genetic disease. So these children will be diagnosed after conception, likely after birth. And so they're not candidates for embryo editing. So that's most children will fall into that category. There are some that have a family history, either because they have a family history, or they went and got their whole genome sequence. And the price of that has come down, so it is reasonable that you could get, your whole genome sequence. You can use those companies like 23andMe and get parts of it sequenced. So in this case, the potential child would be sort of diagnosed before conception, I guess you might say. Okay, so what if they're cystic fibrosis carriers, like we just mentioned? They would probably benefit the most from pre-implantation genetic diagnosis. What if they had Huntington's disease? Now, Huntington's disease is autosomal dominant. That means if you have one copy of the gene, you'll have the disease. So if you have Huntington's disease, you have a 50% chance of passing that on to your kids. But still, you can select for the chromosome that does not carry that trait. So in this case, you have a healthy chromosome shown in blue. You can still select that with pre-implantation genetic diagnosis. So the only use case that I've seen from a healthcare perspective is a diseased autosomal dominant homozygous. So meaning you have both copies of the Huntington's gene. There are a few genetic diseases where you can be really unlucky and you can have an autosomal dominant disease where you have both copies of the bad chromosome. The numbers I've seen, I'm show up here. So for example, Huntington's disease has a prevalence of 1 in 10,000. But that is the second category there, where you have one copy. There are thought to be roughly 350 Americans that have both copies of the diseased gene. So they would be candidates. That's why I drew this arrow very thin. They would be candidates for embryo editing. And that's where a lot of the conversation sort of centers around very small use cases like that. So what about non-therapeutic applications? What other applications might people see in embryo editing? So some are interested in studying development. How does a single cell become a fully functional human being in the course of 9 months? Maybe they want to study what genes are doing in early stages of pregnancy. Or maybe they want to maybe they are passionate about miscarriage and want to understand what causes it to help prevent that. And there's a host of other basic science. But really what you were trying to do when you're doing embryo editing is you're trying to create a gene variant that does not exist in the parents. So in the case of the Huntington's allele, you want to create a healthy one from an unhealthy one. In the case of CCR5, which we just discussed, you want to create HIV resistance when the parents do not have it. There are a lot of other genes that knocking them out creates an interesting phenotype. So one of them is PCSK9. If you don't have this gene, surely, like I said with CCR5, it does something important. We just don't know exactly what that is. PCSK9, people without that gene live healthy lives and don't die of heart disease, pretty much. So your risk of heart disease is much, much lower if you're lacking that protein. If I had to pick one gene to just snap my fingers and knock out of my genome, it'd probably be that one. Or maybe ApoE. So ApoE is also associated with Alzheimer's risk. Now this one, you can't just knock it out. You have to fix it. But there is a risk allele where if you have that copy, you're 95%, or if you have both copies of the risk allele, you're 95% likely to develop Alzheimer's. And if you have the protective version, there's 3 versions. There's the middle case one, and then there's the protective one. And that one, you are much less likely to develop Alzheimer's. But then you kind of get into this gray area, and then you emerge from the gray area into the, height and intelligence. And at some point we have to decide what a distinction between, what is fixing a disease gene. And to be clear, all of my work is in the very obviously disease genes category. No one would say that these genes are, sure, we're enhancing their quality of life, but we're not providing, these kind of Gattaca kind of enhancements. So what are other ways we can do to edit the genome and help improve the lives of patients? So genetic disease. So I've got 4 examples. So I'm gonna do 4 very short stories here. So we go till 12:20 or 12:30. So I'll just kind of go until we run out of time, and then we can continue the discussion after the break. Okay, I will make it to the COVID example, but it's kind of fun. I think I've gotten the impression though that students are done talking about COVID. So all right, so application 1 is HIV. So we'll go back to HIV again because this is the same idea, but now we can do it in people who can sign a patient consent. Right? So the same idea, loss of function of CCR5 gives you resistance to HIV. So the plan is, what if we combine the idea of the embryo editing and the Timothy Ray Brown example into one medicine? So we take bone marrow cells out of patients, fix, or in this case, break the CCR5 gene, and then reinfuse those to see what happens. Okay, so this company called Sangamo is doing just this, and this is kind of a snippet from their clinical trial result. So one thing that they a big challenge with this field though is the drugs for HIV that we have on the market right now are very good. So at day 0, before they start their treatment interruption, they really don't have a lot of circulating HIV. So what they did here is they infused the patients with these edited cells, and then you see this purple area here where it says treatment interruption. They actually stopped their HIV medicines during this to see if this white blood cells that are now circulating would survive HIV. And you can see that there's a kind of a variety of responses. Some patients, like the one with red lines and black dots, didn't do so well. And there was all of them, like the blood cells this is like kind of a percent. The higher they are, the more cells that they have circulating. You can see the orange patient was doing really well at first and then sort of plummeted after the treatment interruption. The patient that performed the best was actually interesting because this is sort of serendipitous. They have an asterisk next to it. That patient was actually halfway delta 32. So one of their chromosomes lacked the CCR5 protein. So they only had half the work left to do to knock it completely out. So this was, kind of considered a semi-success. It wasn't curative. They're trending down. It was unlikely that this was going to be a complete cure, so they put them back on their antiretroviral medication. I don't have the video here, but the next one, but I will send you guys a video of the CRISPR treatment for sickle cell. The sickle cell is probably where you're going to see the most the medicine that's approved that uses CRISPR, well, with maybe one exception in cancer, but where we're fixing the DNA. So in sickle cell, we have the problem is, so we have a mutated gene. This beta globin gene is mutated. When that happens in low oxygen conditions, so when you're exercising, maybe when you're stressed, the red blood cells form these aggregates, these sickled red blood cells. This is a fairly common genetic disease. Many of us may know someone with this disease. And this disease, causes significant pain and sort of premature loss of life. And it's all from one mistaken base pair. So one base pair is the wrong base, and that creates this sickled red blood cell phenomenon that causes so much pain. So if you could correct that one base, you could fix the disease. So which we could take that T and turn it into an A, the disease would disappear. So we can use CRISPR and a homology repair template and try to cut and replace that. But it actually turns out that one of the themes I hope you pick up on the next few examples is that fixing genes precisely turns out to be really hard. The technology is improving a lot and we're getting there, but we're still not quite there yet. An easier thing is breaking genes. We're pretty good at that. There's a gene called BCL11A that switches between fetal hemoglobin and adult hemoglobin. So when you're in utero, oxygen supply, your oxygen comes from a different source. It comes from the placenta from the mother. And so the type of hemoglobin that you have that carries oxygen around is different and has a little different properties. It's still a lot better than, it's not quite as efficient at carrying oxygen as an adult as beta globin, but it's still much better than the sickle globin. And this protein, BCL11A, controls that switch. So if you can break that protein, you can switch back to fetal globin. So this is what this company called CRISPR Therapeutics is doing. And of course, ideally, we would switch it precisely back. We would fix the beta globin gene precisely back to from the sickle cell hemoglobin to the normal hemoglobin. So we're trying to switch from fetal hemoglobin, so showing this gamma, to beta globin. Alpha globin is not mutated, it sort of stays the same. So you can kind of see here, schematically shown what the globin levels are before and after birth. So you can see right at day 0, right when you're born, you're seeing a dramatic switch between fetal globin and beta globin. And so they designed a CRISPR approach to target that enhancer region of that BCL11A protein. So that's, you can just think of it as the breaking the protein. It's a little more in the weeds than that. And by breaking that protein, you get that switchback. So here's the clinical data. So this is the first 2 patients that they infused with this. This is a real clinical trial where they're using CRISPR in people. They codenamed the medicine CTX001. So it's the very first clinical candidate from CRISPR Therapeutics, CTX. So at baseline, you can see in green is beta hemoglobin. It says HbA, but that's adult hemoglobin or beta globin. So you can see that beta globin in green before they start infusing is high, and over time, over the next few months, that green almost disappears. In contrast, you see the HbF, so that'd be the fetal hemoglobin, ramping up over time. And one important thing is that's 15 months post-infusion. So that's 15 months later, it's still sustained. It's not disappearing over time. And patient 2 is a little behind patient 1. This patient was infused later, but you can see the same effect occurring. So, so phenomenal result there, and potentially curative therapy for sickle cell. The next application I want to touch on is cancer therapy. This is another cell therapy application. We're taking cells out and infusing them. And so the exciting technology there is called CAR T cells. So I'm not sure if any of you have heard of CAR T cells before, but this is like a really cutting edge cancer therapy. And yet the idea is T cells go around and kill off cancer sometimes. They sometimes kill bacterial cells and other things that shouldn't be there. So these T cells are part of our body's adaptive immune system. So what if we turned that T cell, what if we trained it to fight cancer? So instead we swap the receptor that T cells use for a different receptor that targets cancers. So that's sort of how T cells work. So you take out T cells, you genetically engineer them to have this new T cell receptor and infuse them. So no CRISPR is involved in that step, but we can use CRISPR and gene editing to make better T cells. And one of the example is the first patient I know that was cured from gene editing. This is a few years back. It was actually using TALEN, which was that predecessor technology I was talking about before CRISPR. This patient, Layla, developed leukemia. I'm sure it's probably a pseudonym, but she developed leukemia, and she wasn't a candidate for some of the chemotherapies. She was so small, and she could not produce enough blood cells to be a therapy for any of the treatments that were available. So they took another patient's T cells, made a new version of these CAR T cells that were lacking the part that would make her body reject those T cells. They cut that part out with TALENs and then infused those into Layla and saved her life. She's 5 or 6 years old now and hasn't relapsed with leukemia. So this success sort of was people hoped it would be kind of a sign of things to come for different curative therapies using gene editing. The third application is hemophilia. I like this application because it's another one of those very clever uses of gene editing. So in hemophilia, you're lacking some proteins that allow your blood to clot and prevent you from bleeding internally, having blood deposits in your joints, or in some cases developing such that anemia that you can be sick or die. There are really good treatments for this. So clotting factor, if you know someone with hemophilia, they can use clotting factor replacements to pretty much remove the disease. There are some rare cases though where that is not an option, and so gene therapy for those patients would be really useful. So these factors are missing, and so if you can replace those factors, that would be beneficial. But the problem is you have to correct quite a bit of the gene in order to get enough. So one idea was, what if we take another gene and steal it and use that to express this factor? So there's a gene that we have in our liver called albumin. It's in our blood. It's one of the highest expressing genes in the liver. So the idea was, what if instead of fixing the factor 8 or factor 9 genes, what if we just put the sequence into this gene, which expresses a whole lot, it makes a lot of protein really fast. We can put that into there, and that would overcome the low efficiency problem that we're having in correcting the protein. And so that's what they did. And they were able to get levels of factor 8 and factor 9 that were so high that they had to bring the dose down in order to reach it. So you see in here, there's The gray area is sort of the therapeutic window that they're aiming for. And so some of their first experiments, they were able to get super physiological levels of this factor expressed in mice. The clinical data is sort of ongoing, but they've seen some good clinical data, not quite as good as the mice, which is always seems to be the case. So they were able to, so you see some of the mice, no patients experience bleeding events, and the factor was still active 61 weeks later. So this is still ongoing, but we'll see more results. One of the interesting things about this, though, is this platform is, you can do this for a lot of genetic diseases. You can put missing genes into this locus and supply lots of replacement genes. So getting back to that early figure I showed you with the 3,000 some odd genes that are mutated, now we can start working on those one at a time with a platform like this. Okay, and the last one is more near and dear to my heart. So it's muscular dystrophy. And I don't know if we finish at 12:20 or 12:30, but I'll shoot through this. So this is a gene that's missing in patients with Duchenne muscular dystrophy. This is dystrophin. And we kind of think of it as the shock absorber of the cell. So in muscles, your muscles are contracting, and it's a lot of physical work on the muscle proteins. This protein binds portions of the muscle and keeps it together. If you're missing this protein, what happens is relatively rare, but it's relatively common in terms of genetic disease. So 1 in 5,000 males will have this. It's X-linked. There's some rare cases where females can have it too. It's typically the mutations result in a complete loss of the protein. Shown schematically there. The phenotype is very severe. Patients die in their 20s. They're wheelchair bound by their teen years. And so it's very severe. There's no treatments that really fix the course of this disease. So we're trying to fix this by breaking it more. So what I mean by that is we can cut out pieces, the mutated pieces of this gene when it's mutated. We can cut out the mutated pieces and fix the protein. And so we do shorten it. We make it a little bit shorter of a shock absorber. That still has a lot of function. So a lot of people have been trying to do this. We used CRISPR to do it. We used CRISPR with 2 sequences to cut out the piece. And the idea is to be able to reframe the protein. So we injected that into mice. And so here's what you see. So on the top is a mouse that does not have dystrophin. You can see some muscle fibers. But what this is looking at a muscle fiber with a cross section. So we're taking a muscle and kind of looking down the muscle fibers. The muscle fibers, you can kind of picture them like a bunch of spaghetti noodles sort of crammed together. And if they light up black in this picture, that means they have dystrophin in them. If they don't, that means that they're lacking dystrophin and that mouse is unhealthy. And so in the bottom here, you can see when we inject CRISPR into the mice, they develop that black staining around the fibers again, showing that we have really good efficacy in fixing the muscle. And so we talked about off targets earlier. What happens if we hit enough targets? So we use next generation sequencing for that. And we do see off target modifications in genes that we don't know the function of. So here's one that's highlighted at OT8, it's off target number 8. And you can see it isn't a perfect match to our RNA, but it still had a significant increase in editing rates. Does it have an impact on the mice? All the mice survived until the endpoint without incident. So the last thing we did is we looked at, did that make the mouse stronger? So we made the mouse basically do a series of 10 squats, is how I like to think of it. So, when you do squats, you do repetitions of a muscle exercise, you sort of lose muscle strength over time. So after the 10 cycles, the WT, that stands for wild types, that's the healthy mouse, loses strength. Our untreated mouse, the sham control, loses muscle strength at a higher rate. And our treated mice maintain their muscle strength to a higher degree. So not quite back up to wild type, but certainly treated. We can show this video maybe when we return, but this is like the hope here that CRISPR therapy can do something like this. I can share this maybe when we get back. I'll pull up the chat here.
Spinal muscle.
Yeah, so I'll look at that. So I'll put the chat here. I can stick around during the break. So I see. Okay. Okay. I'm going to go ahead and off my PowerPoint here.
Wow. Click and clacker back. Wow.
Right.
Crazy, amazing.
Lots of good questions here. Oh,